How many has a background from biology? Right. So little around about half. And if I'm losing you, the other one, or any of you, please stop me and ask questions, because I really want to have everybody along with me for this lecture. So don't be afraid to ask questions. I have many people here that can help me answer the questions, and also you guys with biology background can step in and help me if I'm not able to answer the questions. So talk a little bit about genetic engineering, talking a little bit about GMOs now, and also synthetic biology. I want to show you some trajectories I made. So this is, if you think about conventional breeding, which is not modern biotechnology by definition. We'll talk a little bit about definitions also. What we mean when we say biotechnology, it depends on the viewpoint, of course, and in which setting you are talking. But conventional breeding is really where you select on the phenotype. You know, the bigger plants, the plants that produce more crops, these are the ones you want to bring along and use for the next season. And there was also something called marker-assisted breeding, where you know which genes you want to bring along, so you can use DNA technology afterwards to see that they're actually there. Still, you're not doing anything to the plant, you're just making sure. Marker-assisted breeding. And also in conventional breeding, and this is actually quite important, and you will see why a little bit later. We have something called random mutagenesis, where they normally use radiation or chemicals to make more variants of the plants that they can pick afterwards. They make mutations in the genome, changes in the DNA by using chemicals and radiation. This is not regulated, this is not by definition modern biotechnology. This has been used for a long time, okay? The living modified organisms, or the genetically modified organisms, there are a lot of them, that are bacteria, viruses, and yeast. Normally they will produce additives to food, so medicine, so you will use the product, you won't use the organism for consumption or anything. Also we have the plants, of course, and also the animals. I separated this into two boxes, the single traits, which this will be the crops that have only one genetic engineered trait, like herbicide resistance or insect tolerant. And all of the animals we see now are single traits. But also the plants have evolved quite a bit, or actually the technology has evolved quite a bit, into what we call stacks, where you put more than one trait together into one plant. We'll go more into detail about that a little bit later in a few slides. But the synthetic biology one is really, just a few months ago, well, 6 months ago, I had a similar type of lecture where it was difficult to fill out these boxes. But now this CRISPR technology has really come very strong, and you heard about that from Sarah yesterday, I think. There's a lot of development with CRISPR. So synthetic biology is mainly bacteria and yeast at the moment in big fermenters. And I'm sure Morten will talk more about that if it hasn't already. But also now we see these plants that are made by CRISPR. So I'm putting them under synthetic biology, but I mean, we can talk about that. Should it really be synthetic biology or not? Which is really one of the points also to this presentation definition. We'll talk about that. Keep that in mind. For now, let's keep it under synthetic biology. So the CRISPR plants and animals has been developed already with this CRISPR technology. We know there's a lot of research going on humans as well, human embryos. I'm keeping that medicine thing apart from this. We're not talking about that. We're talking about biosafety connecting to agriculture, to animals, and not human, because that's we're going to be here for a very long time. Also this gene-drive organism, these mosquitoes and stuff like that are being researched at the moment. And I have a question marks at the end, because we've just started here, right? And already there's a lot of things coming up here, so we don't know really where the end is, and that's, of course, that's fine. So, with genetic engineering, what do we mean? Well, at least I mean that there's a direct manipulation of an organism's genes. So, with genetic engineering, what do we mean? Well, at least I mean that there's a direct manipulation of an organism's genes. exception with this random mutagenesis, but at least with genetic engineering, you have an idea what you want to create, and you go in and make a change, a design, engineered change in a plant or product. The methods are really synthetic constructs, used for synthetic purpose, of course, but genetically modified organisms. You have an idea what you want to make, and you take that, call it a construct, a code, and put it into the plant. Or you can also, with the new technologies, you can manipulate existing genes without adding any new DNA, which is a problem for us. It's a challenge for regulation. So, conventional methods, not say too much about this, staying too long here, but hybridization, radiation to chemical mutagenesis, and then modern biotechnology. Because now I want to talk a little bit about definition, technology. What is modern biotechnology? I don't know how many of you, I think perhaps quite a few of you are familiar with the Cartagena protocol. Are somebody not familiar with the Cartagena protocol? No, I've never heard about the Cartagena protocol. Everybody has heard about that. Knows what it is. International protocol governing biosafety under the Convention of Biological Diversity. Very powerful international instrument, right? So they have a definition of modern biotechnology. It means the application, very specific application of DNA, RNA techniques, including recombinant DNA, and also with direct injection into cells or organelles. So you have to have the DNA, RNA component, you have to inject it, or make the cells come together beyond the taxonomic family, meaning that can't happen in nature. So if you do that, then it's modern biotechnology. Okay? So it matters because the Cartagena protocol defines a living modified organisms as an organism that has a novel combination of genes made to the use of modern biotechnology, pointing back to the definition of modern biotechnology. So if you stay within that, that's going to be LMO. If not, if you make a change, there's no DNA there, you don't inject it, outside or inside, which is the debate at the moment internationally, how to make sure that it's going to be regulated or not. So EU, I'm going to say a little bit about EU, because from Europe, has also adopted a definition of GMO, a little bit broader, but has no requirement of this use of modern biotechnology. It just says that if you change the genes in a way that can't happen naturally, then you have a GMO. So, the types of genetic engineering in higher organisms. Well, we have, I'm going to say, well, the GMOs that's on the market today, the BT crops, the herbicide-tolerant crops, the soya, the cotton, the maize, among others, are made by one of these two methods, the ballistic bombardment method or the agrobacterium-mediated transfer. Because you made the construct and you need to get it into the plant, you can use either one of these two methods previously. Now there are a little bit more. All of these actually is a way of making genetic engineering. This is the GMO way, very firmly a GMO if you use these techniques, not so certain that you use these other techniques. A little bit back to that again. So this is an example from, well, it could be any crop, BT crop. Say this is 1A10 for instance, so we'll know about 1A10. This is how it was made. They had an idea what you want to put into there. They made an observation that this bacteria kills this insect. We want to kill this insect, so we take this toxin from the bacteria, the yellow one here, and we need to have some other stuff as well. We need a start signal and a stop signal. There's some technicalities in genetics and some other stuff to make it better. And you make that, and you have made it, put it together, and you optimize the codons. Talk a little bit about optimizing codons to a language that the plant can understand, from the bacterial language to the plant language. And then, well, you have it, but you haven't really it because you need to get it into the plant, right? So one of these two methods I mentioned here. Ballistic bombardment, really just high pressure, shooting it into the plant. Or by the use of this bacteria. You give it to bacteria because that's easy. We've done that in research for years. And the bacteria, this particular bacteria, agrobacterium, if you know plant biology or are connected to crops in any way, I'm sure you heard about agrobacterium. It will put that DNA into the plant. Well, the plant has this very, well, I don't know if it's strange, but when they get DNA into the cell, they have a tendency to take it up, to integrate it into the whole genome, randomly. So you can't be specific with these two methods. And moreover, there are a set of, well, uncertainties, question marks connected with this technology that we can identify on the basis of the technology. We know that when we put the DNA into the cell, sometimes there's mutations and rearrangements, we can't control it, so we need to check afterwards. I'm not saying it doesn't have to be a problem, it's just how it is. Results are unpredictable in the sense that we can't say it must go into this specific spot. And it's the same with the agrobacterium. There's some other sticky things there as well. These plants on the market now made like this, in these methods, have a risk assessment connected to it. They're regulated, and the risk assessment is based on what we know of uncertainties, more or less, the new technologies. Because I'm just going to use this as an example, because the new technologies, when you look at it afterwards, there's no additional genes. CRISPR was talked about yesterday. Basically, you give something to the cells and make a change, and then this thing goes away, and only the change remains. And when you look at it, you can't tell if it's made by this technology or by random mutagenesis. This just happened. You can't tell it by just looking at it. So that means that, and this is the plant's natural process, and that means that there are some voices saying that, well, this methodology just using the plant's own processes, so we don't have to connect it to any risks. So the perception or the idea that certain methods carry certain risks is being challenged with this. So just want to summarize very quickly the types of GMOs we have. We have bacteria, we have yeast producing nutrients and food additives, we have virus vaccines. I'm going to talk more about that later in a separate lecture. And of course the plants, insect resistance, herbicide, some are disease resistant, changed oil content, some vitamins, and also the animals, the fish for growth, particularly the aquabonti. So if we focus on crops, the first generation of GM crops were really single construct, one single gene into the plant, like we saw them on A10. But now more complex constructs has evolved, and they are being combined by a normal process into what we call stacking. It's really when you bring together two different traits, insect resistance, herbicide resistance, you cross them naturally by conventional breeding into one plant, and now you have both of those present. I want to show you an example because it illustrates a couple of points I want to make. So this is what is called MIR162. It's from Syngenta. It has this insecticidal protein similar to BT, kills certain insects. It has a start and a stop signal. It's one copy. It says it's in its native form, meaning it hasn't been changed. It also has something else which is not important when it comes to killing insects, but it is important when they make the plant. And then they crossed in BT11. So in that cross you have these insecticidal proteins still there, and you have this PMI, this monocytic-6-phosphate, yes, it's there. And then you have the POP gene, with its own start and stop signal, which is modified, and then you have a Pri1AB, which is another toxin for killing insects, in the BT11, into one plant. Then 604 was crossed in, and this is an actual example. It's quite an extreme example, I'll give you that, but it's still an example. And then you still have the PUP, the BIP, and you have the Pri1AB. Now you have two copies of this EMI, presumably from this cross 604, which brings in another endotoxin. Now we have a lot of toxins in there already. And then GA21 was crossed in. Now this is a really super complex plant, right? The POP gene the Tri1AB, the VIP, the endotoxin. The PMI is down to 1 copy again, so something has happened there. And then the EPSPS gene, which will allow for tolerance to glyphosate, is from GA21 in 3 copies, actually, not 1, but 3 copies. Actually, it's 3 complete copies and 3 incomplete copies. So that means that the burden for this plant is quite big, and when you cross this with the natural process, you can't design the end result. And the irony of that, in Europe, it's easier to get this approved because all the individual events were approved before, so you can just bring them together. It's easier to get that approval, even though some people, and I, including me, would argue that this is a completely different setup, you know, different process. So this is the classical way of GMOs. But we are also looking at synthetic biology, right? So the new plant breeding techniques, and this is quite, it's a publication from 2011, so it's not since 29. So the picture is quite dramatically different now. But the point is, which still holds true, is that as the year progresses, the research on alternative plant breeding technologies are just skyrocketing. A lot of research is coming up. And on this picture from 2009, CRISPR is not there. That came about in 2012. So it's a very recent development and high adoption. Short commercial break. We at Genok made a report talking about these techniques, looking at biosafety and knowledge gaps. What don't we know, and what do we know we don't know about these techniques? So you can go to that page and download it if you like. If you have problems finding it, please let me know. I was about to bring copies, but I had a lot of papers with me, so I was unable to, unfortunately, but it's available online. One of these techniques that was really talked about just a year back or so are the zinc finger technology. And it's important for a couple of reasons. One of the reasons is that the outcome here is very similar to CRISPR-Cas, or can be similar to CRISPR-Cas. Just very small changes. So what it is really is that you have this setup here, you make this in the lab. This is a recognition sequence. Basically, it's a pair of looking glasses and a pair of scissors. So you give this to the plant. You can decide you have the looking glass what to look for. It goes into the cell. Problem for the plant is when you cut it like this, the plant doesn't know what was there. So you can either give it a template saying this is how it should look like, and then the plant will integrate that here, and you have added a gene. Or you can not give a template, then the plant will just repair regardless, and that will create a mutation. Normally that will stop the protein. If there's something will knock out, this is a good way of doing it. And this will degrade. In the plants that come afterwards, this will not be present, only the change you made. And that's very similar to the CRISPR technology, where also only the change will be afterwards. This was inspired a huge debate in Europe, should this be regulated or not? But there's still no conclusion to that. Because one of the reasons is probably because CRISPR came on so strongly, and they saw the same type of programs going around there, so they would probably not make a premature conclusion to that, which makes sense. Another one I need to talk about is this oligo-directed mutagenesis. I know the names are a bit difficult to remember, but basically what this is we call it ODM for short, where you add DNA, DNA will bind, actually. This is inside the plant, the target, and you will add that thing over there with a gene. So it will go into the plant, bind here because of all these matches. And then there's a mismatch here. And the plant will repair that into what you like to do. So you make a change, and afterward this will also be there. Which is a key point here, because that makes it difficult to regulate. No, you didn't use any proteins, you just use these oligos, and then the plant will say, oh, this is a DNA RNA, this is dangerous, I need to make a change. Or it could also be DNA, but then I need to make a change. And then you have made that. The cisgenesis and intergenesis is terms used in connection with transgenic plants. It was talked quite a lot about, but then it sort of silenced off of it. Basically, it's a GMO made from genes from similar species. The idea behind using specific terms for that, if you take something from another, say you take something from one maize and put it into another maize, or from potatoes, which is quite interesting topic at the moment. You take something from one potato species and put it into another. That's cisgenesis, they say. This term is made because from similar species, from the risk's perspective, the risk should be lower because the genes are from the similar species. Agree with that or not? I would say I'm pretty strong on that. Neither cisgenesis or transgenesis, even if you take it from a species that is similar, you can't make that binormal really. You can't make it go to that exact space where it is being put in with this type of methods. But that is also, to be honest, that's a debate. Many people see this as lesser risks. The scissors and the looking glass has already been deregulated in the US. OGM has been deregulated. And now recently, the CRISPR has been deregulated. This mushroom, made with CRISPR technology, to make it less brown, is deregulated by the US Department of Agriculture. Now there's a reason behind that, and I don't want to go into that. Some definitions again. But some countries are deregulated. In Europe, there is one particular case which is also going to set an example to how this is going to turn out. This is the rape from Cyrus, or it's what we call clear field technology. It's made by ODM. One change in two genes make it tolerate herbicide. When you look at it externally afterwards, you don't know, there's no trace of that engineered process, so you don't know how it was made. If you don't know it, of course, but we know it because they have disclosed that. And actually, these genes were identified by random mutagenesis. They just did randomly, so that if you do something in these genes, okay, then you tolerate this herbicide. So let's only make this change. And you engineer that, you could not tolerate herbicide. But Germany has said that we don't consider this a GMO. You can have this on the market in Germany, inside the EU. Of course, there was a huge protest, there still is a huge protest, and actually, that decision is taken to court. They are now doing a legal battle for that in Germany, whether or not this can be regulated or not. EU, because Germany is a part of the EU, and what EU decides, Germany has to follow, but EU hasn't taken a standpoint yet. They are currently forming a legal interpretation. They will look at the law text already there, the regulations already there, to see if these new technologies should be considered a GMO or not. So there's a lot of attention to the outcome of this, and there's a lot of politics involved, of course. But whatever the EU decides, it's probably going to be a while before it's implemented, because I'm quite sure that this is also going to court. The legal interpretation of the EU Commission, whatever it is probably going to be fought in court, either by environmental organizations or by companies, depending on where they will fall in the decision. So you can see there's no clear-cut answers. Very short about RNAi as well. The RNA is basically what this is. The green stuff is RNA. Remember, the chain of information goes from DNA to RNA to protein. Here, an RNA is made and it binds inside the cell, and then the cell will cut that and silence that gene. So there are concerns for risk assessment, and in 2014 there was a workshop organized by the European Food Safety Authorities, recognizing a lot of biosafety questions connected to RNAi, and also a little bit about how to proceed with that. What is good about this is that this is a start of a transparent process, an open process, a debate, public debate, where people, organizations can get involved and be heard in the process, which is key to getting good acceptance over this assessment. Well, what they can use RNAi for is that you can make a plant with an RNA targeting important genes in specific organisms, because you can make it specific. So in this case, this is specific insects being killed by a specific RNA by design. Yes, no, there's a question about specificity and other insects and off-target effects and all of that we know from risk assessment. But that will always be there, of course. But this is how the technology can work. EU has a definition, and it's quite broad when you read it. It's the application of science, technology, and engineering to facilitate and accelerate the design, manufacture, and/or modification of genetic materials in living organisms. It's a little bit complex, but when you look at it and read it, it sort of encompasses everything, also GMOs into this. Right? So this is not a definition that science would use. Science would have its own definition of synthetic biology, and that's fine. You're not looking from a regulatory point of view, from a biosafety point of view, what can you regulate, what should you regulate and how, right? Science will always challenge this. So the point with this definition is that it is broad, so that it can encompass what we know about GMOs and the experiences we have in risk assessment of GMOs, and include that into the process of supply. So there is a purpose with that, to have it broad. Finally, Cartagena Protocol. If you haven't been to the webpage, you should go here and have a look. There's a separate portal of synthetic biology. The report, which you all have a copy of now, is written by the Secretariat to raise some issues. And the way it works is that there is an online forum. You can click here, list the participants. You can find your participants from your own country, and you can interact with those, of course. So the online forum has going on for some time, and there's different topics that are being discussed. Mainly, how to address the relationship between synbio and biological diversity, what are the operational definitions of synbio, what are the existing national and regulatory instruments, are they sufficient or not to deal with synthetic biology, what should be improved or not. So there's a lot of very, almost a hot topic, but interesting topics going on there. So how does it run that forum? What they did was that they selected a group of experts, and I haven't checked it from which countries. There's a list of those people also on that webpage. You can check if any of you people or somebody you know or from your country are in that expert group. They worked and put together all these topics and put together a report. So this technical expert group gave the report to another body, the SUBSTA. It's a subsidiary body of science. Well, you can read it all. It's a very complicated thing. They came together, at least, and they discussed the outcomes from the ATEC, made big arguments for and against, and that was a really tough discussion. It was in April, now in Montreal, where there are a lot of groups where, well, to be honest, some people don't want regulation and some want regulation. This is the debate, right? This is how it is. So there were groups arguing for less regulation. Now this should not belong under Cartagena at all. And some people say, yeah, it should. And that is really the key thing at the moment. If it falls outside of the Cartagena protocol, if the definition is outside or they will not put it in the Cartagena protocol, that means new audits, new regulations have to be made, and that's not going to happen. Then it will mean that it's going to be deregulated. But if it's inside the Cartagena protocol, then we can build on the risk assessment that we have from GMOs and LMOs. I think it will fall inside. But what happens here in Montreal shows that there is no clear agreement right now, particularly on the definition and on information, genetic information, is really was discussed heavily. Clear sort of borders countries. They will send, and they have an outcome which they will send to Mexico in December 2016, where the meeting of the parties will decide the way forward for bio and regulation. So this, I cannot stress how important this is. If you're concerned about this or want to engage, I think it's very healthy to use your own countries, your own members, delegates, to get involved, have discussion, have meeting, to get a voice heard in that setting. If they decide, no, you're not going to look at it, then it's going to be. I just want to show you the definition that they agree on. And you can see here already that there must be some debate here. It's super long. Many, okay, it's a little bit bigger than what it was before, but this is very long. Further development, synbio is a further development, a new dimension of modern biotechnology. And then to facilitate, accelerate the understanding, but not only the understanding, not only research, design and redesign, manufacture and or modification of the genes, living organisms, and even biological systems. So they are making it broad intentionally. And there's a lot, there are some groups that don't want this, that would like to take this to a much narrower definition. By opening up, basically they're creating a funnel. They want to take as much as possible in and then use their experience to see what should be regulated and to what level. So I think personally that that's a good approach, but of course that's important. If this is going to be regulated and acknowledged, what will we see? Who will use it? Where will it be grown? Well, that is almost impossible to predict. But it will depend on the organisms and of course the usefulness of the product. Are they just going to make the mushroom less brown? I'm not so sure that's going to be all around, but if they want to make something really important in agriculture, saying, then the adoption rate might be much higher. Is it herbicide tolerance and pest management again? Are we going to see this again? Or is it something else? Well, there's a lot of research on this rust, these blight diseases in these crops, wheat, soy, and potato, melting from further on. And I also know the salmon. And there has been some research now where they have made sterile salmon with CRISPR, and it's 100% sterile because it lacks that whole, we call that, the organs for making that happen. So it's absolutely sterile, made with CRISPR now. And it is herbicide tolerance, which is the main trait now. It will change a bit in the future, I'm sure, but now it's herbicide tolerance. And then we have stack traits, more and more complex organisms, all these things put into one plant. And insect resistance is down here. So insect resistance is not so important. This is of course because soybean is being grown so much, right? I will skip that, but where is it being grown? Well, as of 2010 or 11, you can see that the developing countries surpass the industrial countries. So it means that it will be grown all over the world, mainly actually in developing countries. And that means that their regulatory system needs to be in place. It means that you have to get involved with your government, with your regulatory bodies, to have the discussion going, where should it go forward. Already, synbio is a billion-dollar industry. Again, depends on what's being made, how it's being adopted. It's of course not only agricultural products. Bioremediation. If you have spillage, could you make bacteria go out there and use metals and oil, perhaps? Contained use or not is going to be important for risk assessment. Are you just going to keep it in a big tank, or is it going to be out in the environment? It's very different from different risks associated with that. Yeah, you already seen in Europe, herbicide-tolerant rapeseed with this clear technology coming out. Here is undecided, and I just wrote this so to remember to reinforce again that the international process is going to be decisive. I want to end with this article from 2015 in Science. And if you know the authors, you know that these are not anybody. Many of these are Nobel Prize laureates. Odd-Gunnar, these people define modern biotechnology. Also, Jennifer Milner is the first author. She was the one that discovered CRISPR. So what they say, they suggest a proven path forward for genomic engineering and germline gene modifications. These people are mainly concerned with humans and human embryology, but a lot of what they say is applicable, is on the technology, so it's applicable to us as well. So let's just see what in this article, in Science, in March 2015. They recognize, which is important, and it's not something that you would think that it is obvious, but it is sometimes difficult to get that point across. There are limits to our knowledge for human genetics, gene environment interactions, which is important also for crops, right? And animals, gene-environment interactions. Also particles of disease because they are concerned with that. So they recognize that there are limits. And they say, given these rapid developments, it would be wise to begin a discussion that bridges the research community, relevant industries, medical centers, because these are human embryos they are talking about, regulatory bodies, and the public to explore responsible uses of this technology. So what we are saying is that let's think about this for a little bit, and let's start an open debate, an inclusive discussion, so that we can have our boundaries to what we will do and not do with this technology. And that can be applicable, of course, to humans and genetic engineering in humans, which is, of course, ethical difficult, but also should be applicable to other types of research. So I think that with that article, I want to end my presentation. And thank you for your